So first, hi everyone. My name is Jianyu Li, and I'm a PhD candidate at the University of Freiburg. I'm really excited to have all of you joining us today. I want to start by introducing our wonderful speaker today, Dr. Stefan Wolf. Dr. Stefan Wolf got his PhD in theoretical biophysics at the Ruhr University Bochum in Germany. Then he continued his research as a postdoc in several institutions, including University of Wisconsin in the US, the Ruhr University Bochum, Germany, and the CAS MPD Partner Institute for Computational Biology in Shanghai, which was jointly established by the Chinese Academy of Sciences and the German Max Planck Society. He then joined the University of Freiburg in 2016 and completed his habilitation in theoretical physics last year in 2021. His research interest lies at the boundary between physics, biology, and chemistry, and in the rationalization of protein functional motion and the dynamics of intra- and interprotein signal transduction and protein ligand binding, as well as the prediction of underlying physiologically relevant timescales, mainly using multi-scale molecular dynamics simulation on QMMM and QMMM level, employing standard unbiased equilibrium, unbiased non-equilibrium, and biased simulations. And today he's going to talk about breaking computational limits, prediction of drug binding dynamics on orders of minutes to hours. Throughout today's event, you can send any questions into the chat box. If you run into any technical issues, please also send a message in the chat box. And please note that I'm recording the event and will share the recording on my Bilibili channel for others who could not join us today, and so they may benefit from the content. And if you have anything to share with our speaker, also please send them in the chat box and we will address them in the Q&A session. So feel free to add anything you have. And so now I welcome Dr. Stefan Wolf, and now the time is yours.
Okay, thank you, Jenyu, for the invitation again and for the nice introduction. Hello, everybody. So today I'd like to present you our attempts to break computational limits with a special focus on the prediction of drug binding dynamics on the orders of minutes to hours. In the beginning, I'd like to first give you some overview essentially of the underlying physics and the basis of the research that we're working with. So my focus lies in molecular dynamic simulations of biomolecular systems. That means proteins in their interaction partners, such as small ligands, drugs in this case. And that works the following way. Usually, of course, because we are dealing here with molecular systems, we would need to treat everything by quantum mechanics. That is not possible, essentially, because that would be computational way too expensive. So we make the approximation that we can treat these the full system interactions in a way like the atoms would be just small, soft, and interacting spheres, which are connected via springs, so that we can express all interactions via potentials from classical mechanics. And then from this, with this approach, we can essentially derive forces for given atom positions. And now we perform to get an overview of how such a system behaves over time. We perform a so-called numerical integration. That means we take one starting structure, such as a protein here surrounded by water molecules, and now calculate all the pairwise forces of atoms in this simulation box. Then we pretend that we can keep these forces constant for a small certain increment of time, a so-called time step. That would be this delta t here. And now with this approach here, this so-called numerical integration, we calculate the positions of these atoms after this increment of time based on the current positions, the velocities, and the acceleration coming out of the forces. And then when we calculate as well velocities of the system based on the velocities that the atoms have at a given point of time and the acceleration that they experience based on these forces. And so we essentially from this calculate now a new set of coordinates, do this all over again, calculate again a new set of coordinates, and so step through time essentially. And with this, we end up with a step-by-step overview of the dynamics of the system. That's what we call a trajectory. And you see here on the left side, or the right side, Now, an overview of a quite long simulations. This is a protein that we simulated for a couple of microseconds. And you see, for example, here, this part of the molecule is opening and closing again, where you have as well a chemical cleavage site for sugar molecules. And this opening and closing is very important in this case here for this protein to bind around its substrate, chemically cleave it, open again, and release it. Okay, now, what can we do now in case that we have these trajectories? Of course, just we could look at that, but this is quite hard essentially to communicate to other researchers. And as well, it's quite unhandy to really get an overview of what is happening. So usually we are rationalizing these the respective distributions in our system based on so-called free energies. Free energy technically is some a property which is coming out of thermodynamics, and there it is giving you the maximal reversible work that a system can perform. Usually this is related to, for example, the work that a motor can perform. You can apply this as well via statistical mechanics to small to microscopic systems. And if you do then essentially the math or the physics behind this here, you can just see that this free energy essentially is nothing but the logarithm of the probability of the system to be found at a given position. And for example, this X here, this coordinate, that can be, for example, the opening and closing distance here at the mouth portion of this protein that I showed you before. Or what we are more interested in, for example, is the distance between a small molecule drug and its binding site. So if it's large, essentially, the system is unbound. If it is bound, then the distance is short. And the special property here is that, for example, such pharmaceutical important properties like the dissociation constant is then a function of the difference of the free energies between the bound and the unbound state, essentially, so the depth here of the given minima. Physically correct is now here this derivation based on this F here, that would be the Helmholtz free energy. When we're dealing with biomolecular systems, we're living in a constant pressure and temperature environment, and this simply becomes the so-called Gibbs free energy G, which I'm going to use in the following as well. Now, the major part or important part here for the following is that this free energy landscape is a so-called stationary equilibrium property. That means it gives you just an overview of the overall system, but it does not tell you anything about the overall dynamics that the system has, or respectively how long a process takes. So how long, for example, the ligand needs to bind to go from the bound state to the unbound state. That you can calculate as well based on the free energy. So this is here the barrier height between the bound state, for example, and the top of this barrier here, the transition state between the two states. But you need more information about this. So, and this would be this so-called gamma, that's the so-called friction factor. So that means from the free energies, you cannot directly get an information on the respective timescales that the system needs between the two states. Okay. So in case that we want to explore the small, microscopic dynamics and as well the respective microscopic distribution of states, what can we do? In MD simulations, usually what we would do is just we set up the system and let it run and hope essentially that within the given simulation time that we have, we see that the system exploring all states that it has. So for example, for the ligand binding, unbinding case, that would mean that we hopefully see the ligand unbinding, binding again, and see this often enough that we get a well-converged estimate of the probabilities of the system to be in a given spot or at a given distance. And from this, can calculate here the free energies. And again, from the free energies, if we see minima appearing, these are so-called metastable states, that are states that the system remains in, like unbound state, bound state. And the transition states are the important points that we need to understand, because they regulate the kinetics of the system in sorts. Now, okay, that sounds easy, but there's a huge problem that we're facing, which is, again, coming back to this numerical integration is a sequential approach. That means we need to do it over and over and over and over and over again. it is easy to calculate rather large systems, because then essentially we need just to have a lot of computers which work on parallel on those power-wise forces. The larger systems that has been simulated so far are systems on this order of 1 billion atoms. That's quite recent. And again, this is easy if you distribute this over a lot of computers. But for the sequential calculations and therefore assessing large timescales, you need to have fast computers. There's no way how essentially parallelization can help you with this. The crucial point here is that we are limited to a quite small time step in our simulations, because this numerical integration needs to capture the fastest motions that we can see in the system. And in a molecular system, that is usually the bond vibration between an oxygen and a hydrogen atom. And this motion has a frequency of roughly 10 femtoseconds, so 10 to the power of minus 15 seconds. So 10 times 10 to the power of minus 15 seconds. If we want to sample this correctly with this kind of stepwise approach, we need to go down to a tenth of this length. So we are stuck with some step size of 1 femtosecond. That is very small, and the major problem is that the smallest biologically relevant timescale is roughly the microsecond range. So that would be here the 10 to the power of minus 6 second range. So we need to have at least 1 billion iterations that we can go once to one, so that we can see a trajectory length of 1 microsecond. And last but not least, the next point is that these biomolecular systems are not really doing a lot of stuff on these timescales, but essentially you wait for a long time, for example, for such a protein, the protein that I showed you in the beginning, to be closed, and then suddenly it is open again. That jump here just takes a couple of nanoseconds. Then after a couple of microseconds, it closes again. And you see mostly the systems are staying in the respective metastable states. They do not go often into the respective, so they do not switch between these states very often. And for having good estimate, especially on this transition state, you would need to see something like 100 switches between bound and unbound state, or open or closed states. And here, this is one example of this T4 lysozyme protein that I showed you before, with this opening and closing of this mouth portion. And you see that even within the simulation time of 50 microseconds, we just saw that the system jumped 6 times. And that is essentially bad. So because these simulations here took us 2.5 years at the given time, that was roughly 5 years ago. And so we are really hitting here something like a physical border of what we can achieve by means of just simply brute force, unbiased sampling via molecular dynamics simulations. So where this becomes really a problem is, for example, in case that we want to understand how ligands, so small drugs, are binding and unbinding for the respective receptors, because that is a process which is not even happening on microsecond scales, but usually on something like seconds to minutes to hour ranges. And it has been shown actually that understanding these kind of kinetics is very important for pharmaceutical research. Why is that? Well, okay, usually in a pharmaceutical drug design approach, what you do is that you optimize your respective ligand to bind very tightly to its respective receptor or protein where it should act or where it should suppress the signal. That's just essentially based on the way how you can perform these kinds of experiments, like so the small binding assay, molecule binding assays. And there you are essentially then putting together your protein, your ligand, wait a while, see how well it binds there, and you read that parameter out. And that would be then the dissociation constant that you can extract from your measurements. And that, again, as said, is a parameter which depends on the difference in free energy between the bound and the unbound state. That sounds easy, but here's the thing. This approach is only valid in case, or can be transferred to the human body in case that the human body would be an equilibrium system. We're definitely not equilibrium systems, we are non-equilibrium systems. And that's very important, because as soon as we enter thermal equilibrium, actually we are dead. So how does that affect now the pharmacokinetics, so the essentially the dynamics that the system or the ligands have got in our bodies. Usually, if you take up a drug, it's usually oral drugs, essentially they go into your intestines, and from there they go into your blood, diffuse to the respective receptors, bind there, and then perform their actions. It's nice. And then essentially you have your equilibration, local equilibration between an unbound small population, which is then found close to the receptor in your tissue or in the bloodstream. And most of them are going to be bound if you've got a good drug. That is good. And that's essentially how the drug is acting. However, as soon as the compounds are entering your bloodstream, your kidneys and your liver are starting to take out the drugs and to eliminate them from your body so that you excrete them later. So that means this unbound population here is sucked out of the body constantly. And of course, then you are ending up with bound protein, but this bound protein and the unbound state, they are still equilibrating all the time with each other. So you have some constant and in and out switching between the two states. And if this switching is happening fast, then your ligand is essentially can be bound as strong as you want to, but as soon as it becomes unbound, it is directly taken out of the body. That means you end up with a situation maybe where you have a ligand or a drug which is binding very tightly to your receptor, but then is eliminated out of the body quite quickly again as well. So that means you take your pill, you have, for example, a pain relief for 1 or 2 hours, and then afterwards the drug does not work anymore. So for such a drug to act correctly, essentially, you need to take the pill over and over again, maybe maybe each 2 or 3 hours. And that means you have a constant high level of concentration of this compound in your body. And as soon as you have a drug in your body, there's of course as well the possibilities that it binds somewhere else and then acts somewhere where it's not supposed to, and that is causing side effects. And actually, drug side effects are the major constitutions why essentially drugs fail in late clinical stages. That means as soon as you go into into clinical trials with humans, then things are very late stage in the development process, and it's very costly. And then usually pharmaceutical research has costed you a couple of billion dollars or yuan in this case, and you have done everything in vain essentially. So there is some motion in the last decade from the pharmaceutical industry to better understand how you can precisely design drugs which are binding not only tightly, but stay long at their respective binding site. And for this, of course, it would be good to understand the process or the discriminants of a molecule, why it is binding long to a given binding site, and why essentially it stays there. However, again, for being able to perform MD simulations to answer these questions, we would need to be able to perform simulations on scales of minutes to hours, coming back to that. And of course, this is way beyond any kinds of simulation techniques or unbiased simulation techniques or computational hardware nowadays. So how can we essentially go into these timescales if we want to really understand something? Okay. First thing that we can do is that we can try to massively reduce the computational costs which are involved to perform these simulations. And again, usually in MD simulations, we are performing all atom simulations. That means we take some system. This would be a very simple test system, essentially, that we used as well for molecular complexes. That's just essentially a sodium ion and a chloride ion, surrounded by water molecules. And then we will calculate all pairwise forces, and from this propagate Newton's equations of motion. However, what we could do is as well that we could say, well, why not forget about all pairwise forces and now just compute the free energy along, for example, the ion distance, so a parameter which we are interested in, and extract now the free energy of this and somehow use just a calculation of this free energy. for this propagation. And we forget about atoms, essentially. We are coarse graining down our system and not even down to something like just a couple of atoms like you would have in structure coarse graining, but down to just an active field which describes us the dynamics along this distance. Again, the free energy is not sufficient for that, but however, if we could obtain this friction factor that I briefly mentioned in the beginning, then essentially we could essentially describe our full system dynamics along this coordinate of interest based on the free energy and more explicitly the first derivative of it, which is then the so-called mean force. Then the particles would experience some friction based on the velocity that they have, plus some random kicks from the surrounding, from the water molecules here. And these random kicks are coupled by their amplitude, so essentially by their strength, to this friction here as well. And the rest is then just essentially a random fluctuation coming out of a Gaussian distribution. And that would be a so-called Langevin equation. And with this, essentially, we would be able to perform simulations just not based on two parameters. That would mean essentially two vectors. And one would be the free energy dependent on the respective positions along x and the friction along x. And then essentially this whole n body, so thousands of atom calculations, becomes essentially just a table calculation from two numbers that you need to look up to perform this calculation. Now, free energies, again, I introduced, and they are very well understood, essentially what you find behind that. But what is this friction here, especially? So how can friction appear in such a molecular systems where you just have essentially atoms scattering with each other? So the way how to rationalize is that the free energy itself is giving you the mean force that a ligand is experiencing, for example, along the distance from its binding sites at any positions, essentially. And then these fluctuations from the surrounding would be the short time kicks that you receive, for example, by water molecules or ions colliding with a ligand, or as well by fluctuations of the protein at the same time. And because these kicks are so short-lived, we can approximate them as stochastic motions, so random kicks, essentially. And then this mean force here is found in the first derivative of the free energy, and these random kicks are then a function of the friction, or essentially are constituting the friction. So if we had a method how we could extract free energies and friction factors at the same time from an MD simulation, then we could use both these numbers as an input for this Langevin framework and speed up calculations massively. And the way how we do this is by a method that we call dissipation corrected targeted molecular dynamics simulations. This is based again on a quite old approach, which is constant velocity targeted molecular dynamics simulations. Targeted molecular dynamics simulations were developed by Jürgen Schlitter in the 90s. And what you do is essentially that you pull your system with a constant velocity from one state to the other. So that means essentially you pull your ligand out of your binding site with a constant velocity, and you are performing this constraint or applying this constraint to the center of mass of the ligand and the center of mass of the protein, for example. This constraint is acted or calculated by a so-called Lagrangian multiplier. So this comes from Lagrangian equations of the motions of the first kind. And it is essentially a pseudo force that you add to the full dynamics of your MD simulation system to ensure that the ligand is going out of the binding site with a constant velocity. Now, if you integrate all this force over this distance, you end up with a work. And because we are calculating here with a constant velocity, this work is larger than the free energy that we're interested in. This is just essentially a second law of thermodynamics. However, okay, let's see how we could describe that from the side of such a Langevin equation approach. Well, if this force that we calculate from the outside does not act on the free energy, or it's not changing the free energy, and is not changing these fluctuations which are leading to the friction, then we just can add this this force on top of the force experience in this long-run equation framework. And because we are calculating with a constant velocity, there's no acceleration. So essentially this term here on the left side just becomes effectively zero. So the constraint force cancels out all forces or other forces in the system. And if we would now perform a lot of these simulations and calculate mean over them in an ensemble of n simulations, these fluctuations cancel out. We end up essentially just with this with this formula, and then we can kind of identify this mean velocity here just with the constraint velocity that we preset to pull the ligand out of this binding site. If we integrate now this along the distance between the bound and the unbound state, we end up with this expression here where we have a free energy, then the mean work that we need to pull out the ligand from its binding site, and then minus the velocity times the integral of this friction factor over the distance. And that is nothing but, again, this mean work that we have minus a so-called dissipative work. That means a work which is then coming out from the, so which goes essentially into friction and is lost as heat. Now, since the 90s, there exists as well the Jarzynski equality, which is transforming essentially the second law of thermodynamics, which is an inequality into an inequality. And it is written up here. The main problem with this Jarzynski equality is that this mean of an exponential here on the right side, you can, just a moment, excuse me. This equation, this equilibrium, one second. And so the this equality here becomes so this equality here is numerically not applicable to or good to calculate these the free energy that we're interested here on the left side. What we can do is that we can perform a cumulant expansion of the expression and then just essentially stop it after the second cumulant. That is exact in case that the work follows a normal distribution. And then we can just identify this first cumulant here with the mean work that we have in general as well. And then the dissipative work here is nothing but essentially the variance of the work times here a prefactor, which is based on essentially the reverse temperature of the system. Now, what we did then was that we said, okay, let's take this Langevin equation framework that we had on one side and the cumulant approximation from the Jarzynski equality and put that together. And so we can see that essentially the mean, so this variance of work can be expressed as well as the square of the integral over the work, the constraint force fluctuations along our path that we see here. And now with a little bit of math in between, we could come up with an expression for this non-equilibrium work or friction factors, which are now just the integral over the autocorrelation function of these the fluctuations of this constraint force in an ensemble of n simulations. That means we calculate, let's say 100 times simulations where we pull out the ligands from the binding site, calculate the mean force or mean work that we have, and then use this approach here to calculate the non-equilibrium work from our system, and from this then perform a friction correction to our non-equilibrium work to calculate the free energy. So this allows us to remove this dissipative work to directly obtain the free energy from these pulling simulations. And now the first system there where we parameterized this approach with was just this here again, this simple sodium chloride ion system that I showed you shortly before. And what you see here on the right side, where we just pulled apart these the ions from each other, In black, you see the unbiased true free energy that the system has. Here, this is the transition state between the bound and the unbound state. In blue, the non-equilibrium work that you need to pull apart these ions. And now here in red, the respective correct friction corrected free work. And you see that this agrees very well with the respective with the respective unbiased work or free energy, that we're interested in. Now, we were really excited after our results with the free energies here of the sodium chloride system, and said, okay, now let's just use that directly with the protein ligand systems. And then we just saw suddenly that things do not work out. And so you see here now a system that we've been using before as well, or that we use for parameterization. This is the heat shock protein 19. It's a quite fairly common drug target, or for anti-cancer medication. And now here in blue, you see the true free energy that we could derive by another method, free energy method, that would be thermodynamic integration. And suddenly our predicted free energy goes down here drastically to an energy which is so low that actually it would look more like that the unbound state is way more preferred over the bound state. And that's definitely something wrong. So what is happening here? And the problem is that actually ligands can take different routes out of a binding site. So even though we just essentially told the system to push away the ligand from its respective binding site, we saw that we either could see that the ligand then is going out via the top, which you see here on the left side, or it could as well just go forward in the direction of the camera here and then diffuse to the left. And if we have such a path dependency in our input data, we cannot use this simple approach to calculate the friction, because the friction can be different along different pathways out of a binding site. So we needed to develop a method how we can discriminate these kinds of pathways. And for that, we relied on standard methods that we use as well in our lab to discriminate, for example, or come up with discriminate protein, different protein states, conformational states, and to come up with so-called reaction coordinates. That means essentially collective variables, so that are telling us in which, along what kind of coordinates a system is developing. And the advantage with principal component analysis, that is very straightforward. What you, for example, do is, or what we did is that we calculated all the distances between ligands, the ligand and the respective amino acids of the protein. And that what you can calculate is a covariance matrix of these distances over time, and then just simply diagonalize that. And the diagonalization of this covariance matrix is essentially giving you a coordinate system, so a linear combination of protein ligand distances, which is best describing you the respective process. And it tells you as well, so the respective eigenvectors that you yield out of the diagonalization are as well telling you something about in which direction actually the system is going. So if you have a large impact of one amino acid on within this eigenvector, you definitely have a path which is passing by this amino acid. Just now for people who are interested in physics a bit, this covariance matrix, of course, its formulation is only well established for equilibrium simulations, again, because you need to be able to define means. Means are just defined in case that they're time independent. If you have pooling simulations, they become time dependent. But we derived a way how we can essentially calculate these equilibrium covariance matrices out of the respective non-equilibrium covariance matrices, and by this essentially get a good definition of how non-equilibrium principal component analysis works And usually what we do is that we do not perform this approach, but that we calculate the covariance matrix directly out based on the means that we can get out of the non-equilibrium calculations. And what we see is then in this case that we do see pathways appearing in this data set. That's one polling system, polling simulations from another test system. The first main component here, eigenvector, is usually referring to the largest motion, that would just be the pulling coordinate. But you see that along this PC2, the second eigenvector, the system can go either one way. You see this kind of one valley here, which has got one transition state there, or that more pronounced valley down here. And now what we can do is that we can cluster trajectories according to which direction or which with path the ligand is taking out of the binding site, and then perform this non-equilibrium correct friction correction based on the respective path that we have. And this is now giving us good information or good friction corrections as well. And the great thing about this is that, first of all, we can take a look on the respective free energies and as well at friction factors and see what are the underlying mechanisms that define essentially this unbinding. Friction factors here are very interesting as well because they are an information source on all dynamics that you can find within the system, which are not resolved or which are not present within the free energy. Because again, everything that you put into the free energy is everything that you leave out as information of the system, which is then found in the friction factors. And friction factors are usually not calculated because it's really hard, and there are just a couple of few methods which are really can do that. And our method is one of these approaches that really can give you these informations. And this is here another test system where we compared now these the free energies and the friction factors to microscopic dynamics that we could see in the system. In the trypsin-benzaldehyde system, we have trypsin as a blocker, which is sitting here in this kind of peripheral binding site, where you have a strong electrostatic interaction between an aspartate here and the ligand. And what you see is in the beginning that this approach to the transition state is majorly based on the positively charged ligand needing to go away from the negatively charged aspartate, and this salt bridge becomes more and more strained. So it's technically essentially going up here in the free energy is nothing but electrostatics, which becomes essentially strained. And then you see here after the transition state, this along this slope of free energy, you see suddenly that this is the peak where you have the maximum in friction. So the maximum friction is directly here at the turning point of the free energy. And what happens there is that water molecules are rushing into the binding site and can now dampen out the interaction between the negatively charged aspartate and the positively charged ligand. And essentially, so because the water rushes in, of course, it is kicking again the ligand. It is establishing a hydration shell around the ligand and within the binding site. And this establishing of the hydration shell is what is causing the major friction in the system. And then friction goes down to a level where you have essentially just bulk friction. So it just means essentially the ligand is surrounded by water molecules and the hydration, the system is hydrated as well. So we now know as well where are the major sources of friction in the system appearing. Now, coming back to the beginning, so this is nice, but we want to be able how to use that for the calculation of dynamics or timescales. So first of all, already with just using the friction and the free energy as sources of information for an integration of the Langevin equation is speeding up calculations massively. So again, if you have such a small molecule system, you perform simulations, what you end up with is something like 60 nanoseconds on a quite good cluster per day. So that means essentially for a microsecond, you would need to calculate something like 12 days on roundabout. For 1 microsecond with this reduced data set, you just need one single CPU on a standalone modern processor or computer, and you just need 4 minutes for 1 microsecond. And you can go up even with a time step away from 1 microsecond to 5 microseconds because of a couple of advantages in the respective physics there. And we then use this approach directly again to parameterize it with our sodium chloride ion systems, where we have good statistics on the binding and the unbinding kinetics. And by comparing then here the respective kinetics from the fully atomistic simulations with the long-run equation simulations, we saw that actually we are reproducing the kinetics from the fully atomistic systems with a factor of 4. That sounds strange. So essentially we are 4 times slower in our simulations than in the fully atomistic simulations. But remember, going back to the way how you can calculate kinetics, the kinetics are based on the so e to the power of the height of the barrier. That means in your case that you calculate your barrier wrongly by a factor of 1 kT. That's 2.5 kilojoules per mole. That is nothing essentially on the order of error estimates for usual methods. You are already wrong by a factor of e. That means roughly a factor of 3. That means if you have a factor of 4 or even a factor of 10, this is best that you can do essentially with any kinds of methods where you are using the free energy as a source of information to calculate free energy. So we're really good here essentially. We're already with this factor of 4. Now, again, so if we need 4 minutes for 1 microsecond, that's good, but that would require still years essentially to go into this level of seconds to minutes to hour simulations. What can we do there? Well, the big advantage of using fields like free energies or friction factors is that they do not denature if we turn up the temperature, in difference to real proteins or even simulated proteins. And so we came up with an idea which we called temperature boosting of simulations. And that works the following way, that we calculate our friction factors and our free energies at a given temperature that we're interested in, for example, 37 degrees Celsius, so physiologic simulations temperatures, or temperatures where essentially these kinds of, where experiments can be performed, 25 degrees Celsius, 25 or 20 degrees Celsius, and then calculate our fields from via dissipation corrected targeted molecular dynamics simulations at these given simulations with these given temperatures, and then perform long-run simulations at way higher temperatures. And way higher means really we can go up to 1000 or 2000 Kelvin. Perform the respective calculations or integrations at these high temperatures, and then use Cramer's law for transition rates to rescale these kinetics at these high temperatures down to kinetics at the temperatures that we are interested in. And we do this by calculating at a range of different temperatures, and then essentially perform a fit of this temperature boosting algorithm or formula, and then estimate down to the experimental kinetics that we're interested in. And so what we end up with is, in case of the trypsin benzamidine system that I showed you before, there we have binding rates on unbinding rates on the order of milliseconds, and we can recover both binding and unbinding rates with a factor of around about 3. Again, this is really good. And for the HSP90 inhibitor complex, there we have unbinding rates on the orders of half a minute, actually. And we still can recover even the unbinding rate with a factor of 1 to of 20, and the binding rate again of 4. And we require a maximal 1 microsecond of all atom pulling simulations. This is roughly a tenth of what you have, or a fifth of what you need with all or any other methods on the market. So we are quite saving time and computational energy as well, in addition with our respective sampling methods in the beginning. Last but not least, if you do not want to do that, all this will work to characterize one single ligand and want to really scan through a whole library of small ligands. You can do that as well with this approach. And we found essentially that it is sufficient essentially to just take the non-equilibrium work that you need to pull out the ligand, and you can do this at way higher velocity. So usually for this dissipation correction approach, we need something like a pulling velocity of 1 meter per second. So you need to have something like 1 or 2 nanosecond long individual trajectories. If you go up by a factor of 10 with a pulling velocity, so you're ending up with 100 or 200 picosecond simulations, they are very fast. You can just essentially take the non-equilibrium work in the end and essentially use that the average non-equilibrium work in a set of, let's say, 20 or 50 simulations as a scoring function for your respective unbinding rates. And you see here now these are different data sets, and we have a quite good R square between an experimental K-off and the respective non-equilibrium work as well here. This approach, however, is more or less limited a bit based on the set of ligands that you're using. You can just do this as a little caveat for ligands which are chemically very similar. So you cannot go cross chemical scaffolds. And you need to be careful with the electrostatics that you're using for describing your system there as well. So you can end up with quite bad R squares as well. However, pretty much when we compared our approach to essentially standard docking approaches and their predictive power for free energies, you are ending up with roughly the same. So essentially this here is a quite nice and quick and dirty scoring method, which can be used on the same, let's say, mentality like you would use docking for predicting binding affinities. So to summarize all that, so I could hope that I could convince you that with our non-equilibrium work based dissipation corrected target molecular dynamic simulations, which we approximate as DCTMD, together with temperature boosted Langevin equation simulations, we can really reach now these ranges of seconds to minutes to hours that are physiologically relevant for calculating or predicting einbinding rates and binding rates of small molecular ligands. And then as a side note, the non-equilibrium PCA that we developed allows us as really to physically robustly discriminate paths that ligands take out of a given binding site. And just as a little outlook, what we are currently working on there as well is that we do not only use this approach to calculate free energies and friction factors of protein ligand systems and use that for predicting rates. As soon as you have a free energy, you can as well use some external driving force like an electrostatic field and use that, add that simply to the free energy to, for example, calculate then effective potentials that ions are experiencing during transitioning over ion channels. And you can use that to directly calculate then electric currents via our Langevin equation approach. And from this predict current voltage characteristics curves, which are the usual readout parameter in electrophysiologic experiments. So we use that essentially to characterize now ion transport through membrane channels. And secondly, that was more a surprise. These friction factors that we can calculate for via pulling small molecular systems through each other are intrinsically containing some velocity dependence. And that's a very interesting parameter which shows up in the design of, for example, lubricants and technical approaches. That would be a field of rheology and so-called shear thinning lubricants. So we are currently working here together with people from the Fraunhofer Research Institute in Freiburg, who are essentially coming up with approaches how one can predict these velocity-dependent frictions and viscosities based on atomistic structures of lubricants. And last but not least, we are now able to use this approach as well to trigger conformational changes within proteins as well, and from this get an idea on what kind of parts of a protein need to move so that, for example, it can trigger a distance conformational change, and by this get an idea about allosteric changes within a protein. Last but not least, I would like to thank especially my boss, Gerhard Stock, with who together I developed this DCTMD approach. Simon, Miriam, Benjamin, and Matthias are all students which are or have been concerned or working on different aspects of these approaches. Especially funding for these for this approach for our research is coming now majorly from the Forschergruppe 5099 from the German Research Foundation. And if you are interested in reading up on our methods, so here is one transparency with all the respective literature that you would need to look up. And actually, we have published all our codes and evaluation methods as well on our homepage. There you find a link to our respective GitHub repository. With this, I'd like to again thank you for your attention, and I'm happily looking forward to your questions.
Okay, great. Yeah, thank you very much for the wonderful talk. And there are also several questions and messages thanking you for the insights. Okay, and maybe let's start with some questions. The first one is, so on one slide, you showed the you showed the free energy landscape. And the question is, so on that figure, that bound form has lower energy minimal than the unbound form. So what drives the transition from the bound state, the more stable state, to the unbound state comparatively? Okay. Yeah.
Okay, technically, so this is something that we are working on as well to better understand that. Technically, what that is that it is only the thermal fluctuation that this protein ligand complex is receiving. That means essentially, of course, maybe going back to the very beginning, at a given temperature, a molecular, there we go. A molecular system essentially is having a certain velocity range of all atoms. That means essentially the atoms are moving quite strongly. So this is here now a normal molecular system that you see at a given thermal temperature that we have in our bodies. That means these systems are highly dynamic. And even though a ligand is binding very well to its binding sites, it is moving there strongly nevertheless, and it is constantly receiving a lot of kicks, first of all from the protein, and the water molecules are as well from the outside always hitting against the ligand. And sometimes it just happens by sheer luck or just randomly that water molecules just can push themselves by these kicks between the ligand and the protein. And maybe the ligand is at the same time receiving a strong kick from the protein to the outside. And that is essentially driving then just the ligand going out of its initial binding site slightly. And then maybe water can come in, essentially dampening out the electrostatic interaction. And by this, essentially, the ligand is pretty much just kicked out. You can maybe as a more figurative impression, you can imagine the same thing like you go into a very crowded metro station around so high traffic hours, and then trying to get from A to B, and you never can go in a straight line. And even if you push forward very hard, essentially you're going to be hit from the outside as well and pushed around by other people. And it's pretty much the same dynamics there, even though the system we are talking about here a large separation of time scales. Okay, so these individual kicks, they take femtoseconds, so 10 to the power of minus 15 seconds. And unbinding, for example, is happening on the order of, even if it's milliseconds, that means we're separated here by a time scale of 1 billion femtoseconds in between. That means 1 in a billion times of a kick, so the ligand receiving a kick, it's going out of the binding site. So from the molecular perspective, That is very, 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 very seldomly, and there's ages between the binding and the unbinding. But of course, from our microscopic perspective only, that is so fast because we are way bigger than these molecular systems.
Yeah, great, thanks. Our next question is, how long do you need to run a simulation before identifying the collective variables for the calculations? And how do you also identify the right collective variables for the calculation?
Okay, good. First thing, that depends on the respective system that you're interested in. If it's a protein ligand system and you are pushing out the ligands, that's a bit of, first of all, you need to hit the right velocity. That's the one thing that I glance over a bit. So we have the experiments, there's something like a Goldilocks velocity of 1 meter per second. And you need something like, well, 100 to 500 simulations. Again, these simulations are quite short, just 1 to 3 nanoseconds. So you can just put that essentially on the cluster. So we do this here in Tübingen, run them, and usually after a weekend, they're done. And then you need to, well, okay, just essentially, then you need to perform your principal compound analysis. And that is then connected to your second question. Finding the right coordinates, yeah, that's an art, and it's something that we are working on, how to come up with good rules. What is a good reaction coordinate for what process? And that's a not answered question yet. So our experience is that for protein ligands systems, for example, these protein ligand contacts or amino acids distances are quite good discriminant, mostly. Sometimes that doesn't help because it might be that the right collective variable is not containing only a distance, but an angle as well. For example, let's say a ligand needs to go out of its binding site, and then they need to rotate and go out. Of course, such a rotation you do not get by a distance, because this here, this orientation has got the same distances to the binding site like that one. So this is something that we're currently working on with different approaches, how we can include, for example, rotations or internal conformation changes of the ligand. And this is something that we are still concerned with. Sometimes it's even the case that a single hydrogen bond within the ligand can be the discriminant. That was one thing that we will publish soon, where we were quite surprised to come up with that. For protein conformational changes, that's something that we work on as well. There you're mostly stuck with the problem that you need to see enough transitions between, for example, here, an open and closed state of a protein, to be able to get a good statistics of this opening and closing before you can go into searching for the right collective variables. Protein contact distances are quite good starting points, and right at the moment we are usually concerned with using some kind of an iterative approach where we are using the respective principal components to get a better idea. Okay, what individual contacts are important for this transition, and then take a look into the simulations again by eye, just and focusing on these places that we have been hinted on by the PCA, and then from this take it to the next level and get a better idea of what kinds of coordinates are really important. We're sometimes even employing machine learning approaches, but that's tricky, and this is an ongoing research question as well that we're working on.
Yeah, thank you very much. And regarding collective variable, there is a related question from Professor Kamakar. It's about, so work is a path variable. Do you see any collective variable dependency on the pooling process?
Yes, we do. Of course, in each path, essentially, we see has got different work distributions. I'm checking if I usually have something like a slide here for that. So there we go. So this is now just a toy system that I used to work on as well. And the issue is that, for example, you see that each path essentially here, even in this toy model, has got, so I've constructed it in such a way that it has a different free energy. The problem is that each path, essentially, if it has a different free energy even and a different friction, that leads to the problem that we cannot throw all trajectories together, and why we need to discriminate these paths essentially. So because then along each path we can calculate the respective work, and from this then perform respectively for each path this non-equilibrium correction. And then we end up with different paths, and we need to identify the correct path essentially that the system is taking in the non-bias and the equilibrium base as well. And that is actually something that we're still working on, how we can really identify this path. So this is right at the moment we say, okay, it should be the path with the least highest, so with the lowest transition state, because that's essentially the fastest one. But of course, if you have friction then in the play, and then you have a second parameter, which is maybe slowing you down essentially along a path, which is even, so if you have high friction along a path with low free energy barriers, then this might be not so good as having a high barrier and low friction along that. And this is something we still need to work out, or which we're trying to work out what is the right path in this case.
Yeah, thanks. So maybe you have already answered the next question. So any comment on how to deal with multiple unbinding pathways? I'm not sure.
Yeah, that is actually something that we're working on right at the moment. Again, working hypothesis is we take the pathway with the lowest free energy barrier. But again, yeah, we are still working on that, getting a better idea. Usually what we do is at the moment is brute force, take all respective pathways that we find and perform Langevin simulations along that, and then see what is the path with, for example, the slowest, the fastest unbinding rate, and what is the one with the fastest binding rate, and take it from there.
All right, thanks. So again, next question is also from Professor Kamakar. So first, I'm enjoying your talk, thanks. And just a quick question, do you calculate the covariance matrix and principal components from the equilibrium simulation of a single state, or the two states, bound and unbound?
Well, we do this essentially with the biased simulations. Essentially, that means essentially we take the simulations and we put all the information into it. That means we take all trajectories where we pull the ligand out from its binding site. So we have essentially the bound state, unbound state, and everything in between as information. And we take all trajectories and concatenate them to something like one pseudo trajectory where the ligand is essentially just going out and again out and again out and again out. And then the means that go into this non-equilibrium covariance are, of course, the means where the ligand is essentially in the middle. But in case that the ligand is going out once in this direction, once out of this direction, so that we end up with a covariance with a covariance matrix and the respective eigenvectors, where usually the first eigenvector is describing as just the pulling direction. We see this essentially because all essentially so the respective eigenvectors look that way, that all distances all are going up. And then we see, for example, distances in the second eigenvector, distances which need to increase during the simulation or decrease. So that would be the distance, for example, from amino acids which are here, where the ligand is passing by. And from this, essentially, we can get an information which of these eigenvectors are telling us something about which direction the path is taking. The nice thing I have to say is that because we are dealing here with a three-dimensional problem, so the ligand going through 3 dimensions, we are usually ending up as well with only 3 principal components that we need to use to explain these diffusions, and then we can really plot our data or project them down to that, and we do not need to do any kinds of high-dimensional clustering approaches like we would need to have in protein conformation analysis data, which becomes very unhandy, especially when you want to identify paths as well through such a high-dimensional space.
Okay, yeah, thanks, great. And there is a last question. Can metadynamics achieve the same in a shorter simulation time?
From our experiences, so for comparison with the respective literature that I'm familiar with, is that metadynamics needs usually 10 times more simulation time as an input. So we are ending up with something like 5. They need something like 5 to 10 microseconds of input time. Metadynamics has the advantage that they're a bit more flexible when it comes to the respective collective variables, but they need to do some kinds of pathway discrimination afterwards as well. That's a bit hidden in the respective paper by Tivari and Paranello. What metadynamics cannot give you, and that's the important part, is the friction. So usually what you have in metadynamics is the respective free energy profile, and you can estimate the kinetics based on the time that the system needs in the metadynamic simulation from the bound to the transition state. However, what we get for free as well is then the friction factors on top of that. We get binding rates as well at the same time, which is something that metadynamics does not get. And we get a way better convergence essentially of our simulations, because in our high temperature Langevin simulations, we see thousands of transitions. And of course, this is then helping us as well with getting way better convergence of our kinetics, based, of course, on our hope that our free energy estimates and friction estimates are very good. The free energy is converging quite quickly. Friction factors have a quite large error bar, but the friction error goes only linearly only into our estimates of the kinetics, and the free energy, of course, goes exponentially in there. So if our experience is, if the transition state estimate is good, then the friction is just giving you something like a small error on top of that, which is negligible against the barrier for error.
Great. Yeah, thank you. So far we don't have any question anymore. And thanks again for the very, very nice talk. And yeah, thank you all for being here. And there is a last comment from Professor Kamaka, thanking Dr. Stefan Wolf for answering all the questions.
Thank you as well for your questions.
Yeah, thanks. And so there's also another talk from Dr. Adrian Nunes-Alves from Technical University of Berlin. And I just sent the link into the chat. If anyone is interested in this talk, you can get the content from this link. It's also about protein ligand binding kinetics. Yeah. And yeah, so then thank you very much. And thanks for all for being here, and hope you all stay safe, and maybe we see each other next week.